Welcome to the deep dive. We're about to explore something truly fundamental to who we are and, crucially, who every child you encounter will become.
Yeah, we're talking about that intricate, often and a really surprising dance between what we inherit and, what we experience.
And we're kicking things off with a story that honestly sounds like it was pulled straight from a science fiction novel, but it's absolutely real.
It really is. It's the kind of story that immediately makes you lean in and, well, question everything you thought you knew about what makes us unique.
Okay, so picture this. Jim Springer and Jim Lewis. Identical twins.
Right, identical. And they were separated at just 4 weeks old, adopted into totally different families. They didn't see each other again for nearly 4 decades.
4 decades. And when they finally reunited at 39, the similarities were, well, uncanny is putting it mildly.
Uncanny barely covers it. I mean, listen to this. They both worked as part-time deputy sheriffs.
Okay, that's already kind of weird.
It gets weirder.
Yeah.
Both vacationed in the same spot in Florida. Both drove Chevrolets.
Seriously? Chevrolet?
Yep. And they even both had dogs named Toy.
No way! Dogs named Toy, that's just wild.
And get this. They both married women named Betty, divorced them, and then married women named Linda.
Stop it. That can't be true.
It is. And one named his son James Allen. The other named his son James Allen. Same name, just, spelled slightly differently.
That is absolutely mind-blowing.
And the list just keeps going. They both liked math, but weren't keen on spelling. Both enjoyed carpentry, chewed their fingernails, had remarkably similar drinking and smoking habits, suffered from hemorrhoids, even gained 10 pounds at roughly the same point in life, and first got tension headaches around age 18.
Wow. So these similarities spanned everything from hobbies and health to, major life choices and even naming conventions.
They're astonishing.
And it wasn't just the Jims, was it? I remember reading about the Giggle sisters.
Ah, yes. Daphne and Barbara.
Yeah.
Another pair of identical twins reunited later in life. After being reunited, they apparently couldn't stop making each other laugh.
Just spontaneous giggling.
Yeah, this sort of uncontrollable shared trait that their adoptive families had never really seen in them before. They also both apparently avoided stress and conflict, weren't interested in politics. It's fascinating how these core behavioral patterns seem to pop up independently.
So these aren't just one-off anecdotes. These twins were part of a pretty famous study.
Absolutely. They were part of the renowned Minnesota study of twins reared apart, led by Thomas Bouchard and his team, a really landmark study.
What did they actually do in that study?
Well, they meticulously brought together identical twins who share, virtually identical genetic codes, and also fraternal twins who are no more genetically alike than regular siblings.
Oh.
These twins came from all over the world, and they underwent really extensive testing, personality tests, intelligence tests, detailed medical histories, and answered literally tens of thousands of questions about their lives, their habits, their preferences.
So when you have genetically identical twins raised in completely different environments showing such striking similarities, it really forces you to ask the big question, doesn't it?
It absolutely does. The central question of this whole deep dive, are these resemblances purely genetic? Or does the environment we grew up in hold an equally powerful, perhaps even sort of intertwined sway?
And for you, our listener, coming from an elementary education background and clearly interested in child development, our mission today is to go beyond the surface, to really dig into what makes us who we are.
Yeah, we want to unpack the intricate biological beginnings of human development. We'll look at the huge evolutionary forces that shaped us over millennia, the microscopic genetic foundations, the realities of human reproduction.
And most importantly, that dynamic, often surprising interplay of heredity and environment that shapes every single child.
We promise to offer some practical insights, too, things that might help you understand the profound biological influences on the children you'll soon be encountering, or maybe already are encountering, in an elementary classroom.
Because truly understanding a child means understanding their biological blueprint, right? And how it continuously interacts with the world around them. So let's get into it.
Okay.
We've talked about the uncanny similarities of the Jims, really mind-bending stuff. But before we get deeper into genes, let's zoom out a bit.
Okay.
What does something as grand and, ancient as evolution actually have to say about a child learning their ABCs or figuring out friendships in a modern classroom? It seems worlds apart.
It might seem that way, but what's truly fascinating is how natural selection, the concept Charles Darwin laid out way back in 1859, actually laid the initial groundwork for every living thing, including us.
Right, survival of the fittest, basically.
Essentially, yeah. It's the process where individuals best suited to their environment survive and reproduce, passing on those beneficial traits. Darwin saw that organisms tend to reproduce way more than the environment can actually support.
Leading to competition.
Exactly. Competition for resources, for mates. And those individuals with traits that give them an edge in this competition are more likely to survive, and critically, to pass on those advantageous characteristics to their offspring. Over generations, these traits become more common.
Okay, so like an eagle's sharp talons. That's a clear physical adaptation that helps it hunt and survive. How does that translate to human behavior, especially when we think about a child developing?
That's where the concept of adaptive behavior comes in. This is basically any behavior that promotes an organism's survival in its natural habitat.
Okay.
For child development, a prime example, maybe the most fundamental one, is the attachment between a caregiver and a baby.
That strong bond.
It's more than just a nice social bond, though. It's an evolutionarily adaptive behavior. It ensures the infant stays close to a caregiver for essential things like feeding, protection from danger, warmth, all fundamentally increasing the infant's chances of surviving long enough to eventually reproduce themselves.
And for you, as someone heading into elementary education, it's worth considering how these fundamental adaptive behaviors are the very first interactions you might observe.
Absolutely. A baby's innate drive to seek comfort, to respond to a soothing voice, or just to look for connection. These aren't simply learned behaviors.
They're built in.
They seem to be deeply rooted biological predispositions. They lay the essential groundwork for all future social-emotional development. It's like a child's earliest blueprint for social survival, you could say.
Okay, here's where it gets really interesting for me, applying these, ancient evolutionary ideas to how we understand modern human behavior, especially in child development. This is evolutionary psychology.
Indeed. Evolutionary psychology is a relatively newer approach in the field, and it emphasizes how adaptation, reproduction, and that survival of the fittest idea play a really pervasive role in shaping our behavior today.
And fit here doesn't just mean physically strong.
No, not at all. In evolutionary terms, fit means the ability to bear offspring that survive long enough to bear offspring of their own. Passing on those genes is the key.
Right.
David Buss, who's a key figure in this field, argues that just as evolution has shaped our physical features, thumbs, walking upright, etc., it also profoundly influences our decisions, our fears, our social interactions, even things like our mating patterns.
He offers some pretty compelling, though maybe speculative examples, doesn't he? Like the idea that our ancestors' needs shaped some of our current thinking abilities.
Exactly. He and others suggest thinking about hypothetical scenarios from our hunter-gatherer past. For instance, if men were primarily responsible for hunting.
Which required good spatial skills, presumably.
Right. They would have needed superior spatial thinking skills to track animals, navigate terrain, maybe aim projectiles.
Yeah.
So the thinking goes, men born with a sort of innate knack for these skills would be more successful hunters, better providers, maybe more attractive mates.
And therefore more likely to pass on those spatial thinking genes.
That's the idea. Over many, many generations, this could lead to those traits becoming more prevalent in males. Now, it's important to say, critics rightly point out that it's really challenging to prove definitively that this specific historical scenario actually happened like that.
Yeah, complex human traits are rarely down to just one simple explanation.
Precisely. But this line of thinking, applying evolutionary principles to development, directly leads us to evolutionary developmental psychology. This is a branch highlighted by researchers like Bjorklund and Pellegrini.
Okay, so what's the focus there?
They directly apply evolutionary principles specifically to how humans develop. And one of its core ideas, really central, is the extended childhood period in humans.
What's so special about our childhood compared to, say, a chimpanzee's? We take ages to grow up.
We really do. Compared to other primates, humans have both a significantly larger and more complex brain and a much longer childhood. We take far longer to become reproductively mature than any other mammal.
And this long childhood isn't just a byproduct, it serves a purpose.
Evolutionary developmental psychologists argue it's absolutely crucial. This extended childhood, which you, our listener, will witness firsthand throughout elementary school, seems specifically evolved to allow humans the necessary time.
Time for what, exactly?
Time to develop that large, complex brain. And just as importantly, to accumulate the vast array of experiences and learning needed to master the intricacies of our highly complex societies and become competent adults.
So for you, thinking about your future classroom, this implies that the whole structure of elementary school, this prolonged period of learning, isn't just something society cooked up.
It might actually have deep evolutionary roots, potentially during this critical time that specialized modules in our brains, maybe for mathematical knowledge, possibly evolved from ancestral problems like treating goods, or for language, obviously crucial for communication, get extensively trained and refined.
So it's not just about what we teach, but recognizing how our brains might be wired for that long, immersive learning process that childhood provides.
That's a great way to put it. And think about children's natural curiosity, their innate drive to play. These aren't just trivial pastimes from an evolutionary perspective.
They're adaptive behaviors.
Quite possibly. They fundamentally facilitate learning and exploration. The source material mentions an example of an 11-month-old from the Efe culture in Africa trying to cut fruit with a machete, mimicking adults.
Wow, a machete. Okay.
Yeah. But the point is, this isn't just random play. It demonstrates, arguably, an innate drive to engage with tools, to observe, imitate, and problem solve. These are traits that teachers can beautifully harness in a classroom.
Right. With hands-on activities, understanding that a child's urge to explore and manipulate things isn't just restlessness, it's maybe a fundamental adaptive behavior that has served humans for millennia.
Exactly.
But I'd imagine some of these ancient adaptations might not always be helpful in our modern world, right? Like things that were useful then might cause problems now.
That's a really important point. Some behaviors that were highly adaptive for our prehistoric ancestors can actually be detrimental in contemporary society. A classic example is our craving for high-calorie foods.
Like fats and sugars.
Precisely. In an environment where food was scarce and unpredictable, having a strong propensity to gorge when food was available and to crave those energy-dense foods was immensely adaptive for survival. Storing fat was crucial.
But today,
In many parts of the world with abundant, often highly processed food, this very same evolutionary trait can contribute significantly to health problems like the obesity epidemic. It's a powerful example of evolutionary mismatch. What once promoted survival now poses a significant health challenge.
This raises a really profound question then. Are our behaviors just dictated by our biology? Are we just running ancient programs?
That's the million-dollar question, isn't it?
Yeah.
Albert Bandura, who's famous for his social cognitive theory, argued very strongly against what he called one-sided evolutionism.
Meaning the idea that our social behavior is just a product of evolved biology.
Right. He thought that view was too simplistic. Instead, he proposed a more nuanced, bidirectional view.
Bidirectional? How so?
He argued that environmental and biological conditions continuously influence each other in a dynamic loop. It's not just biology shaping behavior, but behavior and environment shaping biology too, over the long term.
That's a fascinating twist. So it's not just a one-way street where biology dictates everything. Our actions, our culture, could actually shape our biology over time.
Precisely Bandura's point. In his view, evolutionary pressures initially led to changes in our biological structures, like, say, the development of hands capable of making and using tools, or brains capable of complex thought and language.
Okay.
This then allowed our ancestors to manipulate their environment in new ways, building shelters, creating tools, developing agriculture, forming complex social structures, inventing language systems.
Creating culture.
Exactly. And in turn, these environmental innovations, these cultural developments, produce new selection pressures. These new pressures then fostered the evolution of even more specialized biological systems, perhaps favoring brains capable of even greater consciousness, more complex thinking, more sophisticated communication. It's a feedback loop.
So biology allows culture, and culture influences biology.
That's the idea. Stephen Jay Gould, a renowned evolutionary biologist, echoed this sentiment. He concluded that biology provides a broad range of cultural possibilities. It doesn't rigidly dictate specific behaviors like aggression or peacefulness. We have the capacity for both, and culture plays a huge role in which path we take.
So what does this all mean for us, and especially for understanding the children we might work with? It sounds like these big-picture evolutionary ideas, while really insightful, are incredibly difficult to actually test or prove through direct observation or experiment.
You've hit on a key limitation. The vast timescales involved in evolution make direct empirical study incredibly challenging, if not impossible, for many questions. We can't run an experiment over millennia.
Right.
So for a more practical and, frankly, effective approach to understanding the mechanisms behind complex traits and behaviors in humans today, we often need to turn to the microscopic world of genetics. Studying specific genes and how they link to traits and behaviors. That's where we can start to unravel the biological nuts and bolts in a more testable way.
That makes perfect sense. It grounds these big ideas in something we can actually observe and measure now. Okay, so let's dive into that microscopic world then.
Let's do it.
Every single one of us started as just one tiny cell, right? Yet somehow packed within that single cell was our entire genetic code. It's mind-boggling. But what are genes, really, and how do they actually work to build a whole complex person?
It is mind-boggling, and the term collaborative gene is really fitting here, as highlighted in David Moore's book, The Dependent Gene. Because the key thing is, genes are not independent operators, just doing their own thing in isolation.
They work together.
They work together, and they work with the environment inside and outside the cell. So, technically, genes are short segments of DNA. That incredibly complex double helix molecule looks like a spiral staircase, right?
All right.
These DNA segments are housed within thread-like structures called chromosomes, located right there in the nucleus of pretty much every human cell.
And their job is?
Their primary job is to contain the instructions for cells to reproduce themselves and, critically, to assemble proteins. Proteins are the fundamental building blocks of our cells, and they're also the crucial regulators that direct countless processes throughout the body. They do the work.
I remember the Human Genome Project, which aimed to map all our genes. Didn't they have a kind of surprising result regarding the number of genes we actually have?
They certainly did. It was a major revision. Initially, scientists estimated humans would have well over 100,000 genes, maybe more.
Based on our complexity.
Exactly. But the actual number turned out to be much lower. The current estimate is around 20,500 genes.
Wow, that's a huge difference. What did that imply?
It was a significant revelation because it showed that humans actually have far more proteins than we have genes. This means individual genes don't operate in a simple one-to-one fashion, like one gene makes one protein.
Ah, so it's more complex. One gene can be involved in making multiple proteins.
Or multiple genes work together, and the process is regulated in complex ways. It underscored that it's a much more intricate, interconnected, and indeed collaborative process between genes and other factors.
That's really surprising. So a gene isn't just like a simple on-off switch for a single outcome. It's more like it's listening to its environment. Is that fair to say?
That's a brilliant way to put it, actually. A gene's activity, whether it's turned on or off or how active it is, what we call its expression, is profoundly affected by its environment, both the internal environment of the cell and the external environment of the body and beyond.
How does that work? What kind of environmental things can affect genes?
Think about hormones circulating in your blood. They can make their way into cells and literally interact with the DNA or the machinery around it to turn specific genes on or off.
And hormone levels can be affected by?
All sorts of things. External environmental conditions like light exposure, the length of the day, the nutrition we receive, levels of stress, even our own behavior can influence hormone flow and other signals within the body.
So stress can physically affect our genes.
Studies have shown that external events, as well as events inside the cell, can excite or inhibit gene expression. For example, research suggests that an increase in stress hormones like cortisol might actually increase DNA damage over time.
Wow.
And perhaps most powerfully for development, we now know that early experiences, things happening in infancy and childhood, can literally alter gene expression in ways that persist and influence later behavior and health outcomes. This is the core idea of epigenetics, which we'll likely get into more later.
This is such a vital connection for elementary educators, isn't it? It fundamentally changes how you might think about potential.
Absolutely crucial. This is perhaps one of the most empowering insights for you, thinking about your future students. A child's genetic potential for, say, intelligence, or for certain talents, or even for certain vulnerabilities, isn't a rigid, predetermined outcome fixed at birth.
It's responsive.
It's profoundly responsive to their environment. Things like good nutrition, a stimulating learning environment, positive and supportive interactions with teachers, reduced stress. All of these are environmental factors that can literally influence how those genes get expressed.
So the environment can help genes reach their potential, so to speak.
Exactly. These positive environmental factors can help a child's genes turn on or express themselves in ways that allow that child to reach their full potential. Think of it like a carefully cultivated garden. The seeds have immense potential, yes, but the quality of the soil, the amount of water and sunlight they receive, those determine whether they simply sprout or truly flourish.
And your classroom environment, the interactions you have, that's part of that vital soil and sunlight.
A huge part of it, you're directly influencing the environmental context in which those genes operate.
Okay, so these incredibly collaborative and dynamic genes, how do they actually get passed from generation to generation? And how do they end up copied perfectly into all the trillions of cells in our body?
Right. Good question. There are three fundamental biological processes at the heart of this story: mitosis, meiosis, and fertilization.
Okay, those terms definitely ring a bell from biology class. Can you
Mhm.
quickly break them down for us in terms of how we get our unique genetic makeup?
Sure. First, there's mitosis. This is how most of your body cells, everything except sperm and egg cells, reproduce. Remember, every normal human body cell contains 46 chromosomes arranged in 23 pairs.
Right.
During mitosis, the cell's nucleus, including all those chromosomes, duplicates itself precisely. Then the cell divides down the middle. The result: two new cells, each containing the exact same DNA as the original cell, with those same 23 pairs of chromosomes.
So that's for growth and repair.
Exactly. That's how we grow from a single cell, how we repair tissues, and how we replace cells throughout our lives. Constant, faithful copying.
Okay, so what's meiosis then? That's different.
Very different. Meiosis is a specialized form of cell division that occurs only to form eggs in women and sperm in men, what biologists call gametes.
The reproductive cells.
Right. During meiosis, a cell duplicates its chromosomes just like in mitosis, but then it divides twice. This results in 4 new cells. And here's the crucial difference. Each of these 4 new cells has only half the genetic material of the parent cell.
Only 23 chromosomes.
Exactly. 23 unpaired chromosomes. Each egg and each sperm cell gets just 1 chromosome from each original pair.
And then these unique gametes, the egg and sperm, come together.
Precisely. That's fertilization. During fertilization, an egg and a sperm fuse to create a single cell, which we call a zygote.
The very first cell of a new individual.
Yep. And in this zygote, the 23 unpaired chromosomes from the egg and the 23 unpaired chromosomes from the sperm combine perfectly to form one complete set of 23 paired chromosomes again. 46 in total. Half come from the mother's egg, half from the father's sperm.
So this is where the incredible, unique combination of genes for each individual comes from. This mixing ensures no two people are exactly alike, even siblings from the same parents, unless they're identical twins.
Exactly right. This whole process, meiosis and fertilization, introduces several incredibly important sources of genetic variability. This variability is hugely valuable for a species because it provides a wider range of characteristics for natural selection to potentially act upon.
So how does that variability actually get generated?
First, during meiosis, when the chromosome pairs separate to go into the sperm or egg cells, which chromosome from each pair ends up in which cell is largely a matter of chance. It's like shuffling 2 decks of cards independently. Plus, before the pairs separate in meiosis, pieces of the 2 chromosomes in each pair are actually swapped between them. This is a process called crossing over. It shuffles the genetic material within the chromosomes, creating brand new combinations of genes on each chromosome.
So you get unique chromosomes going into unique sperm and eggs.
Exactly. This crossing over and the random assortment of chromosomes ensures that every single egg and every single sperm cell is genetically unique. Then when a particular sperm fertilizes a particular egg, the resulting zygote is a truly one-of-a-kind combination of genes from both parents.
And this also explains the difference between identical and fraternal twins, like the gems versus maybe other twins.
It does, perfectly. Identical or monozygotic twins develop from a single zygote that, for reasons we don't fully understand, splits into 2 genetically identical replicas early in development. That's why they share virtually 100% of their genes and can be so strikingly similar, like the gems. Fraternal, or dizygotic, twins, on the other hand, develop when 2 separate eggs are released and fertilized by 2 separate sperm. They're essentially just siblings conceived at the same time, sharing about 50% of their genes on average, just like any other pair of siblings born years apart.
Got it. And you mentioned something earlier about genotype versus phenotype. How does that play into variability?
Right, that's another crucial layer. Even when genes might be similar, people can vary because of the distinction between genotype and phenotype.
Remind us what those are again.
Your genotype is all of your actual genetic material, your complete genetic heritage inherited from your parents. Think of it.
So even if the recipe book, the genotype, is largely the same, the cake, the phenotype, can turn out a bit differently depending on the kitchen conditions, maybe the quality of the ingredients you actually use, like nutrition.
That's a perfect analogy. For each genotype, there's often a range of possible phenotypes that can be expressed, and environmental influences play a huge role in determining where within that range an individual ends up.
Give me an example.
Okay, an individual might inherit the genetic potential, the genotype, to grow very tall, but achieving that full potential height, the phenotype, absolutely requires environmental support, like good nutrition, adequate sleep, freedom from certain illnesses during childhood. Without those environmental inputs, the genetic potential might not be fully realized.
This is such a fundamental concept for you as an elementary educator, isn't it?
Absolutely. It means that even children who might have similar genetic predispositions, like siblings, will inevitably present with unique phenotypes. This is because of the vast genetic variability introduced at conception, and because they experience different environmental influences throughout their development, even within the same family.
It really reinforces the idea that every single child in your classroom is truly unique, developing along their own distinct path, requiring individualized understanding and support.
You got it. Every child is a unique combination of their genetic inheritance and their life experiences.
So if every child in a classroom is truly unique, how do we begin to understand how specific genes actually express themselves to create such diverse characteristics and traits? What are the rules, the principles at play?
Well, we're still learning a tremendous amount. It's incredibly complex. But we do understand several key genetic principles that describe how genes interact to manifest those observable traits, the phenotype. One of the most fundamental is the dominant recessive genes principle. In many cases involving a pair of genes, one gene, the dominant gene, will always exert its effects if it's present. It essentially overrides the potential influence of the other gene in the pair, which is called the recessive gene.
Can you give us a classic example, like maybe eye color or hair color?
Hair color is a good, albeit simplified example. The gene for brown hair is typically dominant over the gene for blonde hair. So if you inherit one dominant gene for brown hair from one parent and one recessive gene for blonde hair from the other parent, you'll have brown hair.
Because the brown gene dominates.
Exactly. A recessive gene, like the one for blonde hair in this case, only exerts its influence and shows up in the phenotype if both genes in the pair are recessive. You need two copies.
So this explains how two brown-haired parents could have a blonde-haired child.
Precisely. If both brown-haired parents happen to carry that recessive blonde-hair gene hidden alongside their dominant brown-hair gene, they can each pass on the recessive gene to their child. If the child inherits the recessive gene from both parents, they'll have two recessive genes, no dominant brown gene to override them, and voila, blonde hair.
Interesting. Okay, what other principles are important?
Another really important principle involves sex-linked genes, often specifically called X-linked inheritance. This occurs when a mutated or altered gene is carried on the X chromosome, one of the two sex chromosomes.
And this affects males and females differently.
Very differently, usually, because males have one X chromosome and one Y chromosome, XY, while females have two X chromosomes, XX.
Right.
If a male inherits an X chromosome that carries an altered, perhaps disease-causing gene, he doesn't have a second X chromosome with a potentially normal copy of that gene to compensate. Therefore, males are much more likely to express or show the effects of that X-linked condition.
So females with their two X chromosomes are more likely to be just carriers, meaning they have the gene but don't show the condition themselves.
Exactly. Females have that backup X chromosome. If one carries the altered gene, the other X usually has a normal copy, which is often sufficient to prevent the condition from developing. So females are frequently carriers. They carry the gene and can pass it on, but typically don't show any signs of the disorder themselves.
Can you give an example of an X-linked condition?
Sure. Hemophilia is a classic example, affecting blood clotting. Another one relevant for educators is Fragile X syndrome, which is a common cause of inherited intellectual disability, and can also involve features like learning disabilities or a short attention span. It's significantly more common in males because it's X-linked.
Okay, that makes sense. What else?
Then there's a really fascinating phenomenon called genetic imprinting. This is where the expression of a gene actually differs depending on whether it is inherited from the mother or the father.
Wait, the same gene behaves differently based on which parent it came from?
It's complex. But essentially, during the formation of sperm and eggs, a chemical process marks or imprints certain genes, effectively silencing one copy of the gene pair in the offspring. So for some genes, only the copy inherited from the mother might be active, while the copy from the father is silenced, or vice versa for other genes.
Is that normal?
Yes, imprinting is a normal and important part of development for a number of genes. But when this imprinting process goes awry, if the wrong gene gets silenced or activated, it can disrupt development and lead to certain disorders, like Beckwith-Wiedemann syndrome, which involves overgrowth.
Wow, genetics is complicated, and I imagine most of our characteristics like height or intelligence aren't determined by just one or two genes following these simple rules.
You're absolutely correct. The vast majority of human characteristics are far more complex than simple dominant recessive patterns or single gene effects. Most traits are polygenically determined.
Polygenic, meaning?
Well, meaning they're influenced by the interaction of many different genes, possibly hundreds or even thousands, each contributing a small effect. Think about something like height. It clearly runs in families, suggesting a genetic basis, but there isn't a single tall or short gene. It reflects the combined influence of many genes interacting with each other, and of course, with environmental factors like nutrition.
So things like intelligence, personality, even susceptibility to common diseases like cancer or diabetes, those are polygenic too.
Absolutely. These complex traits and conditions develop as a consequence of intricate interactions among multiple genes, sometimes referred to as gene-gene interaction, working in concert with numerous environmental factors. This complexity is what makes human traits so incredibly diverse and nuanced. No two people, except identical twins, have the same combination of gene variants influencing these traits.
It really drives home how unique each child is at a biological level.
It really does.
Okay, so we've talked about the usual ways genes work, but sometimes things don't go according to the typical genetic script, right? This leads to what we call chromosomal and gene-linked abnormalities.
That's right. And these variations can significantly impact a child's development. It's very likely that you, as an educator, will encounter children in your classroom who have some of these conditions, so having a basic understanding is helpful.
Can you maybe elaborate on the difference between chromosomal abnormalities and gene-linked ones, and perhaps give us the most common examples relevant for an educator?
Definitely. So chromosomal abnormalities typically involve issues with entire chromosomes, either having an extra copy, missing one, or having a significant structural change like a deletion or duplication of a large segment.
Okay, problems with the whole chromosome structure.
Exactly. The most well-known example is probably Down syndrome. This is usually caused by the presence of an extra third copy of chromosome 21 instead of the usual pair.
And what are the characteristics?
Children with Down syndrome often have distinctive physical features like a rounder face, a flattened skull profile, an upward slant to the eyes, maybe a protruding tongue, shorter limbs. More significantly for development, there's typically some degree of intellectual disability, ranging from mild to severe, and often delays in motor development as well. It occurs in about 1 in every 700 live births, and the risk increases with the mother's age.
Are there other common chromosomal issues?
There are also sex chromosome abnormalities. For instance, Klinefelter syndrome occurs in males who have an extra X chromosome, making them XXY instead of XY. They might be tall, have undeveloped tests, and sometimes enlarged breasts. They may experience learning disabilities. Turner syndrome affects females who are missing an X chromosome, XO, or have part of one deleted. They are typically short, may have a webbed neck, are infertile, and often have difficulty with mathematics, though verbal ability is often quite good.
Okay, so those are issues with whole chromosomes. What about gene-linked abnormalities? Are those different?
Yes. Gene-linked abnormalities are caused by mutations or alterations in specific individual genes, not entire chromosomes. Over 7,000 such single gene disorders have been identified, though thankfully most are rare.
Can you give an example that really highlights the nature-nurture interplay we've been talking about?
Absolutely. A crucial example, especially for understanding that interplay, is phenylketonuria, or PKU. This is a recessive gene disorder, meaning a child needs to inherit the faulty gene from both parents.
And what does it do?
Individuals with PKU cannot properly metabolize an amino acid called phenylalanine, which is found in many protein-rich foods. If it's left untreated, phenylalanine builds up to toxic levels in the body,
Causing
severe intellectual disability and hyperactivity. It used to be a major cause of developmental delay.
But I've heard PKU is highly treatable today, right? This sounds like a perfect example of environment impacting a genetic outcome.
Exactly. This is where it perfectly illustrates the power of the environment interacting with genes. PKU is now easily detected at birth through a simple blood test, which is standard newborn screening in many places.
And the treatment?
The treatment is remarkably effective, a special diet that strictly limits phenylalanine intake. This diet needs to start very early in infancy and be maintained. If a child with the PKU genotype adheres to this diet, the excess phenylalanine doesn't build up.
And the outcome?
Intelligence develops normally, or very
Yeah.
close to normally. So the same genotype, the genetic predisposition for PKU, can lead to vastly different developmental outcomes, vastly different phenotypes, depending entirely on the environmental context. specifically the nutritional environment provided.
That's incredible. It powerfully demonstrates that genes are not always destiny. A timely environmental intervention can completely change the trajectory.
Precisely. It's a cornerstone example of GXE interaction. Other examples of gene-linked disorders you might hear about include cystic fibrosis, sickle cell anemia, which impairs red blood cells and is more common in people of African descent, Tay-Sachs disease, and Huntington's disease. The key message is that these genetic variations are part of the spectrum of human development.
That PKU example is incredibly empowering. So, for educators, what's the big takeaway when thinking about children who might have genetic conditions?
It's a profoundly hopeful message, I think. Genes are not destiny in a rigid sense. Even if an individual carries a genetic predisposition for a disorder, or perhaps a vulnerability, other genes or crucially developmental events and environmental factors can sometimes compensate or mitigate the effects.
Like the PKU diet.
It's exactly like the PKU diet. It underscores the immense potential for intervention and support. This growing understanding of genetics is also why genetic counselors play such a vital role today. They are professionals trained to help families understand genetic risks, the odds of conditions occurring, and strategies for managing or offsetting potential genetic effects. They provide crucial information and support.
And for you, as an elementary educator, this knowledge is incredibly valuable, isn't it?
Immensely. You will likely encounter children with various genetic conditions, whether it's Down syndrome, Fragile X, something rarer, or even just subtle predispositions. Understanding the biological basis, acknowledging the condition exists, but critically, understanding that the environment can profoundly impact how that genetic potential unfolds, that's empowering.
How so?
It allows you to provide tailored support, to use adaptive strategies in your teaching, and most importantly, to create a truly nurturing, supportive, and stimulating classroom environment. Your role is to help optimize each child's development, irrespective of their specific genetic predispositions, helping them learn and flourish to their fullest potential.
That's a really positive way to frame it. Okay, let's shift gears slightly. Moving from the microscopic world of genes to the very beginning of life itself. What tools are available these days to help prospective parents understand fetal development during pregnancy and navigate reproductive choices?
Yeah, advances in biological knowledge and technology have really opened up a lot of possibilities here, offering many choices for prospective parents. Sometimes these choices raise complex ethical dilemmas, but the tools themselves provide incredible insights.
So what are some of the main ways doctors can look at how a baby is developing before birth?
We can broadly think about prenatal diagnostic tools in two categories: screening tests, which indicate if there's an increased risk for a condition, and diagnostic tests, which provide a more definitive yes or no answer.
Okay, start with the screening tests. What's common?
One of the most common and probably most familiar is ultrasound sonography. This can be done quite early, often around 7 weeks, and then usually again later in pregnancy. It uses high-frequency sound waves, totally painless and safe, to create visual images of the fetus in the womb.
What can you see with ultrasound?
Yes, excellent for checking the baby's growth, confirming the due date, looking for major structural abnormalities like issues with the heart or brain, like microencephaly, an abnormally small brain, determining the number of fetuses, twins, triplets, and confirming the baby's sex. And as I said, virtually no known risks.
What if the ultrasound shows something potentially concerning?
If an ultrasound indicates a possible abnormality, doctors might recommend a fetal MRI. This uses powerful magnets and radio waves to create even more detailed images than ultrasound. It's particularly useful for getting a clearer picture of complex issues, especially involving the baby's brain, spinal cord, chest, or other internal organs. Still considered very safe.
Okay, so those are mainly about looking at the baby's structure. What about checking for genetic or chromosomal issues specifically?
Right. For that, we move towards diagnostic tests, often done if screening suggests a higher risk or if the family has a history of certain conditions. Two main invasive tests are CVS and amniocentesis.
CVS?
Chorionic villus sampling, or CVS. This is typically performed earlier in pregnancy, usually between 10 and 12 weeks. A technician takes a tiny sample of tissue from the placenta, the chorionic villi, which has the same genetic makeup as the fetus. This sample can then be tested directly for hundreds of genetic and chromosomal abnormalities.
And amniocentesis?
Amniocentesis is usually performed a bit later, between 15 and 18 weeks. In this procedure, a doctor uses a thin needle, guided by ultrasound, to withdraw a small amount of the amniotic fluid that surrounds the fetus in the womb. This fluid contains fetal cells that can be cultured and tested for chromosomal abnormalities like Down syndrome and other genetic disorders.
Are there risks with CVS and amnio?
Yes, Because they are invasive procedures, both carry a small risk of miscarriage, although the risk has decreased over time with improved techniques and is now quite low, roughly 1 in 200 to 1 in 300, maybe even lower depending on the center. They offer highly accurate diagnoses, but the timing and small risk mean parents have to weigh the pros and cons carefully. CVS allows for earlier diagnosis, while amnio might provide slightly broader testing possibilities later on.
Are there less invasive ways to test for genetic risks?
Yes, definitely. There's maternal blood screening, often called the quad screen or similar names now. It used to be the triple screen. This is done typically between 15 and 20 weeks. It simply measures the levels of certain hormones and proteins in the mother's blood. Abnormal levels can indicate an increased risk, not a diagnosis for conditions like Down syndrome or neural tube defects like spina bifida. If the risk is elevated, diagnostic tests like amnio would then be offered.
And I've heard about even newer blood tests.
Absolutely. On the cutting edge is non-invasive prenatal diagnosis, or NIPD, sometimes called NIPT testing. This is really revolutionary. It works by isolating tiny fragments of fetal DNA that naturally circulate in the mother's bloodstream.
Wow, from just a maternal blood draw.
Exactly. From a simple blood test on the mother, labs can analyze this fetal DNA to screen with high accuracy for common chromosomal abnormalities like Down syndrome and even determine the baby's sex, sometimes as early as 9 or 10 weeks. It's also being used to detect certain single gene disorders inherited from the father.
That sounds amazing, much safer than CVS or amnio.
It is much safer, virtually no risk to the pregnancy. It's rapidly becoming a standard screening tool. However, it's still primarily a screening test. A positive result usually still requires confirmation with CVS or amnio for a definitive diagnosis. And of course, the ability to get this information so early and non-invasively also raises complex ethical considerations for families regarding their reproductive choices.
That makes sense. So for you, our listener, thinking about elementary education, understanding these early diagnostic capabilities is valuable, right?
I think so. It helps you recognize that some children entering your classroom might have had prenatal diagnoses, maybe they received early interventions because of it, or maybe their families went through significant emotional journeys or made difficult decisions based on this information. This background knowledge can just inform your sensitivity to the diverse experiences families bring with them and maybe help you be better prepared if a child has specific learning needs related to a prenatally diagnosed condition.
Right. It adds another layer to understanding the whole child and their family context.
Okay, so moving on slightly. Not all paths to parenthood are straightforward, and many couples face challenges conceiving naturally. What options exist for couples experiencing infertility?
Yeah, infertility is more common than many people realize. It's generally defined as the inability to conceive a child after 12 months of regular intercourse without contraception. It affects a significant number of couples. Estimates range from 10 to 15% in the United States.
And the causes can be varied. It's not always one partner.
Absolutely. The causes are varied and can stem from issues with the woman, the man, or sometimes a combination of factors, or even be unexplained. For women, common issues might include problems with ovulation, not releasing eggs regularly, blocked fallopian tubes preventing the egg and sperm from meeting, or uterine issues. For men, it could be related to producing too few sperm that aren't motile enough, don't swim well, or blockages that prevent sperm release.
So what options are available for couples facing these challenges? It's a huge area. Over 2 million couples seek help for infertility every year in the U.S. alone. The options can range quite a bit. Sometimes surgery can correct an underlying problem like unblocking tubes. Hormone-based drugs can be used to stimulate ovulation in women. And then there's the realm of assisted reproductive technologies, or ART.
RT. That includes IVF.
Yes, by far the most common and well-known ART technique is in vitro fertilization, or IVF. In vitro just means in glass, referring to the lab dish.
Can you briefly explain how IVF works?
Sure. In a typical IVF cycle, the woman takes medications to stimulate her ovaries to produce multiple eggs. These eggs are then retrieved surgically. In the lab, the eggs are combined with sperm, either the partner's or a donor's, in a dish to allow fertilization to occur.
And if fertilization happens?
If fertilization is successful, the resulting embryos are cultured in the lab for a few days. Then one or more of the healthiest-looking embryos are transferred directly into the woman's uterus, hoping implantation will occur and lead to pregnancy.
Is IVF usually successful?
Success rates vary quite a bit. They depend heavily on factors like the cause of infertility and, very significantly, the mother's age. Success rates decline noticeably for women in their late 30s and especially over 40.
And are there any particular outcomes or things to know about babies conceived via IVF?
Well, one notable consequence of fertility treatments in general, including IVF, is a significant increase in the rate of multiple births, twins, triplets, sometimes more. This happens because often more than one embryo is transferred to increase the chances of success. So about 25 to 30% of RT pregnancies result in multiple births, which naturally carries higher risks for both mother and babies compared to singleton pregnancies.
What about the health or development of IVF babies themselves? Are there long-term differences?
That's been studied extensively. Some large meta-analyses have shown a slightly increased risk of low birth weight for babies conceived via IVF, both twins and singletons, compared to naturally conceived babies. However, the reasons aren't fully clear. It might be related to the underlying infertility, the IVF process itself, or the higher rate of multiples. But importantly, when researchers look at broader developmental outcomes, the picture is very reassuring.
Reassuring how?
A key longitudinal study by Golombuk and colleagues followed children conceived through IVF, adopted children, and naturally conceived children into adolescence. They compared their socio-emotional functioning, things like peer relations, self-esteem, school functioning.
And what did they find?
They found no significant differences between the groups on these important measures. While more research is always ongoing, especially on very long-term health, this is a very positive finding for parents who've used IVF and for educators who will have IVF-conceived children in their classes. It suggests these different paths to parenthood don't inherently lead to different developmental outcomes in these key psychosocial areas.
That is really reassuring. Okay, besides IVF, adoption is another really crucial pathway to building families. And you mentioned earlier it's changed quite a lot over the years.
It truly has. Adoption, the social and legal process of establishing a parent-child relationship between individuals who are unrelated by birth, has seen significant shifts.
How so?
Well, particularly in the last 30 or 40 years in Western countries, fewer healthy, non-Latino white infants are available for domestic adoption, partly due to factors like increased access to contraception and abortion, and more single mothers choosing to raise their children.
So who is being adopted now?
This has led to significant changes. There's been a large increase in adoptions from other countries, international adoption, more adoption of children from different ethnic groups within the U.S., transracial adoption, and very significantly, more adoption of children with physical or mental health challenges, or children who have spent time in the foster care system due to neglect or abuse.
And the adoptive parents are more diverse, too.
Absolutely. Adoptive parents today are much more diverse than in the past. They include single adults, gay and lesbian couples, older adults. Societal views and agency policies have become much more inclusive. There are generally no income requirements, for instance.
Okay, so what about the outcomes for these adopted children? Given the diversity of their backgrounds and experiences, is it generally a positive path for them?
It's definitely a complex picture with some important nuances to understand. Research consistently suggests that the timing of adoption matters significantly. Children who are adopted earlier in life, especially as infants, tend to have more positive outcomes and fewer adjustment difficulties overall. Those adopted later, say after age 10, often face more challenges, which makes sense given they may have experienced more instability or trauma prior to adoption.
And compared to non-adopted children, are there differences?
On average, yes. Studies, including large meta-analyses, show that adopted children and adolescents are somewhat more likely to experience psychological problems, like anxiety, depression, externalizing behaviors, and school-related problems, like learning disabilities, than their non-adopted peers. They're also more likely to use mental health services.
That sounds potentially challenging for them and their families.
It can be. However, it's crucial to put this in perspective. While these group differences exist on average, they're generally small. And there's a very important positive side to this story.
Okay, what's the positive side?
The vast majority of adopted children, including those adopted at older ages, transracially and internationally, adjust effectively and grow up to be well-functioning adults. Their parents overwhelmingly report considerable satisfaction with the adoption. Longitudinal studies following adoptees into young adulthood have found no differences in self-esteem or rates of antisocial behavior compared to non-adopted peers.
So the early challenges don't necessarily persist.
Not necessarily, no. And some research has even highlighted positive characteristics. For example, one study found adopted adolescents were less withdrawn and engaged in more pro-social behavior, like being altruistic, caring, and supportive towards others, compared to their non-adopted peers. And critically, adoption consistently leads to far better outcomes for children than remaining in long-term foster care or institutional environments. Children adopted from institutions often show significant catch-up growth in cognitive development.
That's really important context. So what about parenting adopted children? Are there specific things adoptive parents need to consider?
For the most part, the core principles of effective parenting are exactly the same as for biological children. Being supportive, caring, involved, being a good communicator, helping the child develop self-control, all those fundamentals apply.
But there are unique aspects, too.
Yes, adoptive parents definitely face some unique circumstances. Key recommendations often include explicitly recognizing the differences involved in adoption, communicating openly and honestly about the child's adoption story from an early age, showing respect for the child's birth family, even if the circumstances were difficult, and supporting the child's natural curiosity and search for self and identity as they grow older, which often involves thinking about their origins.
And these needs and conversations change as the child grows, right? So thinking specifically about the elementary school years, which is our listeners' focus, what should teachers be aware of?
Yes, the conversations and needs definitely evolve. In early childhood, maybe around ages 4 to 6, it's considered a natural time to begin talking about adoption in simple, concrete terms. Experts strongly advise against secrecy, as finding out later can create significant psychological risks.
Okay, so openness from the start.
Right. Then, during middle and late childhood, those elementary school years, say 7 to 11 children, typically start showing more interest in their origins. They'll begin asking more complex questions. What did my birth parents look like? Why was I adopted? Do I have biological siblings?
And how might they feel about it?
It's very normal for them to have mixed feelings during this time, perhaps feeling grateful for their adoptive family, but also curious about, or even sad or angry about, their birth family or the circumstances of their adoption. Ambivalence is common and perfectly okay. A key point from the source material is for parents, and implicitly supportive adults like teachers, to avoid trying to make the child's life seem too perfect, as this might discourage the child from expressing difficult or negative feelings about their adoption. They need space for the complexity.
That's really insightful. So, as an elementary teacher, you'll likely have adopted children in your class. Understanding their unique identity journeys, the potential for complex feelings, and maybe some of the challenges they might be slightly more prone to, like learning disabilities or just needing to process their adoption story, it allows you to be more empathetic, right?
Absolutely. You can provide appropriate resources if needed. Perhaps ensure classroom libraries include books featuring diverse family structures, including adoption, and most importantly, foster an inclusive classroom environment where all family types are recognized and respected. And having open communication with the adoptive parents within appropriate professional boundaries can be incredibly beneficial for understanding the child's specific needs and tailoring support effectively.
That makes a lot of sense. Okay, this brings us right to the core of it all, the classic nature versus nurture debate. After everything we've discussed, from the incredible similarities of the Jim twins to the microscopic world of genes and environmental influences, can we ever truly separate the influence of heredity from environment? It really seems like they're just completely intertwined, not really opposing forces at all.
You've hit on this central challenge and really the modern understanding. The field that specifically tries to unravel the relative influence of heredity and environment on individual differences in human traits and development is called behavior genetics.
Okay, behavior genetics. And what's its main goal?
It's important to be clear here. Behavior genetics doesn't really aim to tell us how much a specific individual is affected by nature versus nurture for a particular trait. That's almost impossible to disentangle. Instead, it seeks to understand what accounts for the differences we see among people in a population. To what extent do people differ because of differences in their genes or differences in their environments, or some combination of the two?
Right, it's about explaining variation in a group. And how do they go about studying something so incredibly complex? How do they try to tease apart genes and environment?
To study these complex influences, behavior geneticists primarily rely on two main kinds of studies: twin studies and adoption studies.
We talked about twin studies earlier with the Jims. How do they work more formally?
In twin studies, researchers compare the behavioral similarity of identical monozygotic twins with the behavioral similarity of fraternal dizygotic twins. The logic is this: identical twins share virtually 100% of their genes, while fraternal twins share only about 50% on average, just like regular siblings.
So if identical twins are more similar for a trait than fraternal twins are?
It suggests that heredity plays a significant role in influencing that trait. For example, If identical twins are much more similar in their level of reading ability or extroversion, or even risk for a certain condition compared to fraternal twins, it points towards a genetic contribution to those differences.
Are there any problems with that logic?
Yes, there's a potential complication. Critics point out that identical twins' environments might also be more similar than fraternal twins' environments. Parents might treat them more similarly, dress them alike, they might share more friends, etc. This equal environments assumption is debated, and researchers try to account for it, but it does complicate the interpretation sometimes.
Okay, so what about adoption studies? How do they work?
Adoption studies offer a different way to try and separate genetic and environmental influences. In one type, researchers look at whether the characteristics of adopted children are more similar to their adoptive parents, who provide the home environment but share no genes, or more similar to their biological parents, who contributed their heredity but generally didn't raise them.
Ah, so if the child resembles the biological parents more for a trait, it suggests genetics. And if they resemble the adoptive parents more, it suggests environment.
That's the basic idea, yes. Another form of adoption study compares adopted siblings, genetically unrelated but raised in the same home, with biological siblings, genetically related and raised in the same home. These studies, using different approaches, help researchers estimate the relative contributions of genes and environment to variations in particular traits.
So for you, thinking about your classroom, when you observe striking similarities or maybe surprising differences among your students, even between siblings.
Behavior genetics provides a framework for understanding that both their inherent genetic predispositions and their diverse environmental experiences are playing a role. It really highlights that no two children, not even siblings raised in the same family, have truly identical experiences.
Right. That leads perfectly into this idea that the interaction is way more complex than just nature versus nurture. Sandra Scar, a prominent behavior geneticist, talked about how our genes actually influence the environments we end up in, didn't she?
She did indeed. It's a really powerful concept. Scar proposed a model of heredity-environment correlations. She described 3 fascinating ways in which our genetic makeup can actually shape or influence the types of environments we are exposed to throughout our lives. It's not just that the environment acts on our genes influence where we are and what happens to us.
That sounds like our genes aren't just passive recipients, but they're actively involved in creating our experiences. That's a different way of looking at it.
It really is. The first type she described is passive genotype environment correlations.
Passive? How does that work?
These occur because biological parents, who are genetically related to the child, provide both the genes and the rearing environment for the child. The environment the child is passively placed in often matches their genetic predispositions because it matches the parents' predispositions.
Okay, give me an example relevant to an elementary classroom.
Sure. Think about reading ability. Parents who are genetically predisposed to be good readers and who enjoy reading are likely to pass on some of those genetic predispositions to their children. And because they enjoy reading, they're also likely to provide a home environment rich in books, read to their children often, and model reading behavior.
So the child gets both the genes and the book-filled environment.
Exactly. The child passively inherits this environment that encourages reading, which fits well with their potential genetic predisposition for reading skills. It's passive because the child didn't choose that environment. It was provided by the parents. In your classroom, this child will likely arrive at school with a stronger foundation and maybe a greater love for reading, which you as a teacher can then beautifully build upon.
Okay, that makes sense. What's the second type?
The second type is evocative genotype environment correlations. These occur because a child's genetically influenced characteristics actually elicit or evoke certain types of responses from their environment, from the people around them.
Whoa, so a child's own innate traits can literally change how others react to them.
Precisely. Think about temperament. An active, smiling, sociable baby generally receives more social stimulation, more smiles, more cooing, more interaction from caregivers and others than a passive, quiet, fussier baby might. Or consider a child in school who is cooperative, attentive, and seems eager to learn.
They probably evoke more positive, encouraging, and instructional responses from the teacher.
Exactly. Compared to maybe a child who is more distractible, disruptive, or withdrawn. The child's genetically influenced tendencies evoke different reactions from the environment. In your classroom, a naturally curious or outgoing student might evoke more engagement from you or perhaps from their peers, which in turn shapes their ongoing learning experience. Conversely, A very shy, withdrawn child might unintentionally receive less direct social interaction, which could inadvertently reinforce their quiet nature. It's a feedback loop again.
Fascinating. Okay, and the third type.
The third type is active genotype environment correlations, often referred to as niche picking.
Niche picking, like finding your spot.
Yes, that's exactly what it means. This occurs when children actively seek out environments, or niches, that they find compatible and stimulating. Environments that fit well with their own genetically influenced abilities, interests, and personality.
So they're not just passively receiving or evoking environments, they're actively choosing them.
Right. As children get older and have more autonomy, this becomes increasingly important. For example, children who are naturally outgoing and sociable will actively seek out social contexts, joining clubs, playing team sports, initiating interactions with peers. Shy children, conversely, might actively avoid large social gatherings and prefer solitary activities.
Or a musically inclined child might beg for piano lessons or join the school band.
Perfect example. Or a child with strong athletic abilities might spend hours practicing basketball or trying out for teams. They're actively selecting environments that align with their talents and interests. In elementary school, you might see this when a child with a natural aptitude for art always gravitates towards the art center during free choice time. Or a child with strong gross motor skills consistently chooses physically active games during recess.
So, as an educator, recognizing these inherent niches that children seem drawn to can help you. What?
It can help you provide diverse learning opportunities and maybe even guide children towards positive niches that allow them to explore and develop their individual strengths and talents. You can support their choices while also encouraging them to try new things.
And the relative importance of these three types of correlations changes as a child grows, right? Scar suggested that.
Yes, she did. Passive correlations are generally considered more common and influential in infancy and early childhood because during those years, adults largely control the child's environment. The child is mostly receiving the environment the parents provide.
Makes sense.
But as children mature and gain independence, going to school, choosing friends, developing hobbies, they increasingly extend their experiences beyond the family's direct control. They become more active in selecting their own environments. So, active or niche-picking correlations become more prominent and influential during later childhood, adolescence, and adulthood.
These correlations really highlight how children aren't just passive blobs shaped by their surroundings. They're active agents who shape their own experiences, partly based on their genetic predispositions.
Exactly. It's a much more dynamic view. And a teacher's role, then, can involve both recognizing the kinds of responses children might evoke and also helping them find and navigate positive niches that support their individual strengths and address their needs.
Okay, this all suggests that even within the same family, sharing the same parents and house, siblings can turn out incredibly different. We all know families like that. Why is that, according to behavior genetics?
That's a great question. And it leads to another really important distinction that behavior geneticists make. The difference between shared and non-shared environmental experiences.
Shared and non-shared environments. Okay, break that down.
Shared environmental experiences are, theoretically, those aspects of the environment that siblings living in the same home have in common. This would include things like their parents' personalities and general parenting style, the family's socioeconomic status, the intellectual orientation of the home, e.g. lots of books versus few, the neighborhood they live in, the school they attend, though maybe not the same teacher. These are the factors assumed to make siblings growing up together more similar.
Okay, that sounds straightforward. So what are non-shared experiences then? They must be the key to the differences.
Precisely. Non-shared environmental experiences refer to a child's unique individual experiences, both within the family and outside it, that are not shared with a sibling, and thus are thought to be major contributors to the differences we see between siblings.
What kind of things count as non-shared?
It's a broad category. It includes things like parents potentially interacting differently with each child, maybe favoring one, being stricter with another, responding differently based on the child's own unique personality. That evocative correlation again. It also includes having different peer groups, different friends, different hobbies or activities, experiencing unique life events, like an illness or accident that affects only one child. And crucially for our discussion, having different teachers at school each year.
Wow. So even seemingly small differences in how parents treat siblings or having a different best friend or a particularly inspiring or uninspiring teacher, those all count as non-shared environment.
Yes, exactly. And behavior geneticists like Robert Plowman have argued quite strongly, based on twin and adoption studies, that these non-shared environmental experiences actually account for most of the environmental influence on differences in traits like personality and interests.
More than the shared environment, more than the family atmosphere they supposedly share.
Yeah.
That's what much of the research suggests, surprisingly. Plowman argues that the shared environment, the factors siblings have in common, actually accounts for relatively little of the variation we see in many psychological traits once genetic influences are factored out. Think about it. Siblings raised in the same home often have remarkably different personalities, interests, and even outcomes.
Right.
Plowman also suggests that heredity itself influences these non-shared environments through those evocative and active correlations we just discussed. For instance, a child genetically predisposed to be athletic might actively seek out sports environments and friends who share that interest, creating a very different set of non-shared experiences compared to their less athletic sibling in the same house.
This brings up that controversial theory by Judith Rich Harris in her book, The Nurture Assumption, where she basically claimed parents don't really make much of a difference in shaping their kids' personalities, arguing it was all genes and peers. What's the current thinking on that? Is she right?
That book certainly stirred up a lot of debate. Harris argued forcefully that shared family environment has little impact, and that peers are the main environmental shapers of personality outside of genes. While her work highlighted the importance of peers in non-shared environments, the vast majority of developmental psychologists, including behavior geneticists like Plowman, disagree with her conclusion that parents don't matter significantly.
So parents do still matter, despite the emphasis on non-shared environment.
Absolutely. Critics of Harris point out that she may have underestimated the complexity of peer influences and largely dismissed the profound impact parents have, especially in early childhood. For example, parents play a critical role in selecting neighborhoods and schools, which influences peer groups. They model behavior, provide emotional support, teach values, and their overall involvement, communication style, and relationship quality are consistently shown in countless studies to be vital for children's development, well-being, and outcomes. So while genes and non-shared experiences, including peers, are undeniably important, parents remain a central, crucial part of a child's developmental equation.
That's good to hear. So for you, as an educator, this distinction between shared and non-shared environment is profound, right?
It really is. It means that while siblings might share a home address, their actual lived experiences, how their parents interact with them specifically, the unique friends they make, and very importantly, the different teachers they encounter year after year contribute significantly to their unique developmental paths.
So my interactions with each student, the specific dynamics within my classroom peer group, the individual learning opportunities I provide, these are all potentially powerful non-shared environmental experiences for that child.
Exactly. You are a significant source of non-shared environmental influence, capable of shaping a child's development in ways that might be quite different, even from their own siblings who pass through the same school. It highlights the impact an individual teacher can have.
Okay, this is all painting a picture that's incredibly dynamic. It's definitely not a simple one-way street where genes just dictate everything or environment just molds a passive child. What's the latest thinking on how genes and environment truly deeply interact? Is there a model that captures this complexity?
Yes, the current perspective really emphasizes this dynamic interplay. It's often referred to as the epigenetic view, a term strongly associated with the developmental psychologist Gilbert Gottlieb.
Epigenetic view, what does that mean?
This view states that development is the result of an ongoing, bidirectional interchange between heredity and the environment. It's not a static relationship where genes set limits and environment fills in the rest. Instead, genes and environment are constantly collaborating and influencing each other throughout development.
So I like that analogy you used earlier. Like, genes are light switches, but the environment is the hand that can flick them on or off, or maybe even install a dimmer switch. It's a constant two-way conversation between our biology and everything we experience. Is that capturing the epigenetic view?
That's a fantastic analogy. That captures the essence of it really well. From the very moment of conception and continuing throughout the entire lifespan, environmental factors are constantly interacting with our genetic material.
What kind of factors?
It's incredibly broad. It includes exposure to toxins or nutrients during prenatal development, levels of stress experienced by the mother or the child, the quality of caregiving, learning experiences, social interactions, exposure to illness, the encouragement or discouragement a child receives. All of these things can subtly or significantly modify genetic activity.
So they don't change the DNA sequence itself, but how the genes work.
Exactly. They don't change the letters of the genetic code, but they can change how readily a gene is read or expressed, effectively strengthening or weakening its influence, turning it on in one context and off in another. This ongoing dynamic interchange between genes and environment produces every aspect of who we are: our intelligence, our temperament, our height, our abilities, our vulnerabilities. It's truly a continuous dance where heredity influences experience and experience influences heredity's expression.
And within this view, there's the concept of gene-X environment, or GXE interaction. How is that different?
GXE interaction is a specific focus within that broader epigenetic view. It looks at how specific measured variations in our DNA, like having a particular version of a specific gene, interact with specific measured aspects of the environment to influence a particular outcome or phenotype.
Okay, can you give us a concrete example of this kind of specific GXE interaction that might relate to a child's well-being or development?
Sure. Now, a classic example comes from research on a gene called 5-HTTLPR. This gene is involved in regulating the neurotransmitter serotonin, which plays a role in mood. Studies found that individuals who had a specific version, the short allele of this gene, have an elevated risk of developing depression.
Okay.
But, and this is the crucial GXE part, only if they also experience a significant number of stressful life events like maltreatment or major losses.
So the gene alone doesn't cause depression. It requires the environmental trigger.
Exactly. In individuals with the long allele of the same gene, the link between stressful life events and depression was much weaker or non-existent. The gene variant created a vulnerability or sensitivity to the environmental stressor. It's the interaction that elevates the risk.
So that's a powerful illustration.
Another study found a similar interaction. Adults who had experienced parental loss as young children were more likely to show signs of unresolved attachment issues later in life. But again, only if they also carried that short version of the 5-HTTLPR gene. The long version seemed to confer some resilience against the negative effects of that early environmental adversity. These studies clearly demonstrate that a genetic predisposition often requires a specific environmental context to be expressed.
This concept of GXE interaction really underscores the critical role of the elementary school environment, doesn't it? It's not just background noise, it's an active player.
It absolutely does. As an elementary educator, internalizing this concept is invaluable. A child might walk into your classroom carrying a genetic predisposition, perhaps for anxiety or a learning difference or maybe even a remarkable talent. But the environment you create in that classroom.
The support, the stress levels, the teaching methods.
Exactly. A supportive teacher, a low-stress atmosphere, instruction tailored to their needs, positive peer interactions. These can act as crucial environmental factors that either trigger or mitigate the expression of that child's genetic loading. Your classroom is a dynamic part of this complex environmental equation, constantly interacting with each child's unique biology and shaping their developmental trajectory in real time.
Wow. Okay, so let's try to bring this all together. To answer our initial question about the Jim twins, and really about all of us, what's the conclusion? Is it nature? Is it nurture?
The clearest conclusion from decades of research is that the contributions of heredity and environment are not additive.
Yeah.
You can't simply say a trait is, 50% nature and 50% nurture, as if they're separate ingredients you just pour into a bowl and mix.
Right. It's not like baking a cake where you can measure out the flour and the sugar separately. It's much more like a complex chemical reaction where the ingredients transform each other.
That's a much better analogy. Genes produce proteins throughout the entire lifespan, in many different cellular and external environments. And crucially, whether they produce these proteins and how actively they do so depends significantly on how harsh or nourishing, as the source material puts it, those environments are.
So genes provide potential, but environment shapes the reality.
That's a good way to think about it. The emerging view is that complex behaviors likely have some degree of genetic loading, maybe a propensity, a tendency toward a particular developmental trajectory. But the actual development, the unfolding of that behavior or trait, requires interaction with a complex environment. And that environment is incredibly broad. It includes everything we traditionally think of as nurture, like parenting, family dynamics, schooling, neighborhood quality, culture. But also biological encounters like nutrition, exposure to viruses or toxins, even random events occurring at the cellular level.
This all sounds like we're shaped by these incredibly powerful forces, genetics and environment, constantly interacting. It can almost feel deterministic. Are children, are we, completely at the mercy of our genes and our experiences?
That's a natural question to ask. While a genetic heritage and our environmental experiences are certainly pervasive and powerful influences on development, the consensus is that humans are not completely at their mercy. We have agency.
Agency, meaning we have some control.
Yes. As the psychologist David Myers concluded in a quote referenced in the material, we are both the creatures and the creators of our worlds. We are undoubtedly products of our genes and environments shaped by forces beyond our complete control. But the stream of causation that shapes our future also runs through our present choices, our intentions, our goals.
So our minds, our consciousness, our ability to plan and choose, that matters too.
It matters immensely. Our hopes, our goals, the expectations we hold for ourselves and others, the effort we put in, the choices we make every day, these significantly influence our future paths and outcomes. We aren't just passive puppets of biology and circumstance.
And this, for you, our listener, heading into elementary education, is perhaps the most empowering message of all from this deep dive, isn't it?
I truly believe it is. While children arrive in your classroom with their unique genetic blueprints and come from diverse, complex home environments that you may have little control over, the classroom itself is a powerful domain. It's a space where positive environmental influences can be intentionally, thoughtfully applied.
So my teaching, the relationships I build, the learning environment I create, they're not just side notes, they're actively participating in that child's development.
Absolutely. You become a significant author in that child's developmental story. Your actions can provide the nourishing context that allows positive genetic potentials to flourish and potentially buffer against vulnerabilities. You can help children navigate challenges, discover their strengths, and help them fulfill their boundless potential, whatever their starting point. It's a position of incredible influence and responsibility.
Wow. What an incredible deep dive into the biological beginnings of who we are. We've gone from those just unbelievable similarities of twins raised apart right down to the microscopic dance of genes and environments.
Yeah, and the clear takeaway is that development is this truly dynamic, ongoing conversation between nature and nurture. They're not opponents, they're collaborators, for better or worse.
And understanding this, it isn't just, academic curiosity. It's really about appreciating the unique, complex journey of every single child who walks into your life or into your future classroom.
Exactly. Knowing that genes themselves are collaborative and that their expression is so profoundly influenced by the environment, especially by supportive, nurturing conditions, it just highlights the immense power of providing those conditions.
So for you, as an aspiring elementary educator, this knowledge really does become a kind of superpower, doesn't it? You're not just teaching reading or math.
No, you're becoming a vital part of each child's unique developmental environment. You have the potential, through your actions and the classroom climate you create, to influence gene expression, to help kids find positive niches, to provide those crucial non-shared experiences that really contribute to their individual developmental trajectory.
Okay, so here's a final thought to leave you with, something to really mull over as you continue on your path in education.
Given the profound impact that the environment has on how genetic predispositions actually unfold, consider this. How might the intentional, the compassionate, the informed design of every single elementary classroom, and indeed every single interaction within it, become one of the most powerful forces we have for shaping not just minds, but the very biological expression of potential? How can we best use that power to foster resilience and true flourishing in every child we meet?